Okay, our next speaker is Dr. Taylor Sundby. And Dr. Sundby, it's notable that every year Giant Food gives a special award called their Next Gen Award. And the purpose of the Next Gen Award is to basically support the bridging time of somebody going from being either a very senior fellow or very junior faculty into getting a little more funding and establishing their own independent career. And Dr. Sundby fortunately received one of these awards from CCF in 2020. So we congratulated you then, and now we get to see the fruits of what that young investigator, that Next Gen Award did for you. Dr. Sundby is now at the NCI as an assistant research physician, so he's moving up the ranks. And he's going to give a talk entitled Cell-Free DNA Fragmentomics. Going to tell us what fragmentomics are. Distinguish between benign, pre-malignant, and malignant peripheral nerve sheath tumors in neurofibromatosis type 1. Dr. Sundby.
Perfect. Thank you. No, and it's true that this project and my junior career really did start with that CCF funding, so it is my pleasure to talk to you guys about the use of circulating biomarkers to distinguish between different types of peripheral nerve sheath tumors and the cancer predisposition in F1. I do have some patent filings related to cancer biomarkers, but otherwise no disclosures. So just to set the stage for this group, and I know that we have a diverse group of clinicians, scientists, as well as community and survivors, so please ask questions if I don't define anything along the way. But NF1 is the most common cancer predisposition. It affects about 1 in 3,000 individuals. And the leading cause of mortality in this population is an aggressive soft tissue sarcoma called a malignant peripheral nerve sheath tumor, or an MPNST. These have an abysmal 5-year overall survival, and that's largely because they're resistant to both chemotherapy as well as radiation therapy. So really, early surgical resection is the key to treatment. But this is difficult in the setting of NF1 because these MPNSTs tend to arise from within, or at least near, these benign precursor tumors, plexiform neurofibroma, and this pre-malignant lesion called an atypical neurofibroma. And as you can see, these tend to arise during early childhood, early adolescence. MPNSTs tend to come in usually the 3rd decade of life, though with some genetic predispositions or lesions, it can occur a little bit earlier. And so the difficulty for early surgical resection is early detection. And you can see from this that it can be really difficult to distinguish between these tissue types. So you can imagine that if you're doing a standard tissue biopsy where you're putting a needle into the tissue, you can be a few millimeters up towards the top right corner, and you're going to get the diagnosis of a peripheral or of a plexiform neurofibroma and miss the MPNST that's there until it might be metastatic or more symptomatic. This is also very difficult by imaging. Partly because this tissue does intermingle, but also because most patients have multiple plexiform neurofibroma. So being able to monitor those and pick which lesion is the most likely to transform is difficult, and whole body MRI at this point is really mostly used in the research form. And so what we hypothesize is that we can use something called cell-free DNA to accurately distinguish between MPNST and plexiform neurofibroma. And so cell-free DNA are naked fragments of DNA that are shed from tissue, cancer tissue. It also comes from your healthy tissue. And importantly, these are in your circulation, so we can grab it from your blood. You can actually get it from tears, any other biofluid. And they recapitulate all of the genetic lesions, so anything that we can get from the genetics of a tumor, most anything, we can also get from the cell-free DNA. And so this first part of the study, which was funded by CCF, I used copy number variation, so the number of copies of genes, to distinguish between malignant and peripheral nerve sheath tumors. And so you can see here that the molecular pathogenesis going from plexiform neurofibroma to MPNSTs is relatively well defined. There's stepwise changes in the genome that lead to this. One of the sort of low-hanging fruits is that as it becomes malignant, you have increased aneuploidy, which is just a fancy way to say that you have more copy numbers of various genes. And so you can see in this right inset that the MPNSTs tumors on average have 438 copy number alterations, and this is from tissue, whereas plexiform neurofibroma and atypical have much less, so 9 and 23. And so in this first project, we took samples from patients with healthy volunteers, patients with plexiform neurofibroma, and patients with MPNST. We took blood, separated the plasma from that blood, and from that plasma, we're able to get these fragments of DNA called cell-free DNA. We sequenced those and looked at copy number changes. And there's a global score that we can use called tumor fraction, which gives an overall marker of the genomic instability, how much aneuploidy is there in each sample, and we correlated that with clinical data. And what you can see is that this actually did relatively well at separating MPNST from plexiform neurofibroma. So that first line is saying that in someone who has MPNST, who hasn't received treatment, radiation, surgery, or chemotherapy, we are able to accurately distinguish MPNST with 86% accuracy from a single sample. If we have multiple samples, we can do that with 89% accuracy. And importantly, in this chart, you can see that the blue are the standard diagnostics right now, anatomic MRI and image-guided biopsy. And our sensitivities and specificities are relatively comparable to these. Anatomic MRI might require sedation in these patient populations. And image-guided biopsy is obviously invasive, and again, for the reasons that we saw in that picture, can have a high positive predictive value. If it says you have MPNST, you probably have MPNST, but a low negative predictive value, meaning you might have missed by a few millimeters, and you say you have plexiform neurofibroma, but there's still an MPNST lurking there. And I apologize, just put a pin in the chart on the right, but notice that I talk about size-selected and non-size selected, and that's what most of this talk today is going to be talking about when we talk about fragmentomics. And then I'm going to breeze through this, but importantly, these green boxes represent areas from MPNST tissue that we expect to see copy number alterations. The red are copy number gains, the blue are copy number losses, and that's from aggregate cell-free DNA data. So you can see that the genomic alterations that we see in the cell-free DNA map to where we would expect to see it based on tissue. Importantly, plexiform neurofibroma don't have many of those copy number alterations, and that healthy in the bottom right is nice and diploid, meaning 2 copies of each gene. It's all gray, almost all gray. And so from this first study, we were able to say that cell-free DNA can accurately distinguish MPNST from plexiform neurofibroma. We saw that cell-free DNA captures and recapitulates the MPNST genomic landscape. And though I didn't show the data, we see that cell-free DNA correlates well with clinical findings. So in fact, changes in tumor fraction can precede radiographic or imaging changes of progression or new metastases. And again, this all was funded by CCF at this point. And so that was published in 2021. And since then, we've worked on validating this data. And what you can see here is the MPNSTs are in red. Everything else, so we have our healthies, our NF1 is this NF1, patients with NF1 but no peripheral nerve sheath tumors, our plexiform and our atypical ANUPs, all of those are relatively flat and separate from our MPNST. But what you may also notice is that MPNST is the only one that really stands out from these, right? So you would be unable to use copy number alteration to separate from any of the entities on the left-hand side. And these are clinically important entities because we treat them differently clinically. And so plexiform neurofibroma tend to be treated with MEK inhibitors or just observed. Atypical neurofibroma, most institutions would resect because we want to try to get rid of it before it has the chance to transform. But importantly, because it's not malignant, you can use less morbid surgical, narrow margin resections. MPNSTs, we have to use wide oncologic resections. They tend to have a lot more morbidity attached to them, and many of these patients are metastatic at diagnosis, and so they require neoadjuvant chemotherapy or radiation therapy. So what we hypothesize is that we could look at these fragmentomic profiles, which is just the sexy term that we apply to it to say we look at these little fragments of DNA and we look at different characteristics from them, to distinguish between everything that was on the left of that chart that did not fall out with copy number alteration. And so this is from the first paper, but this is what led to this hypothesis. And so you can see here on the x-axis, we have the length of the cell-free DNA fragments, shortest on the left-hand side, longest on the right-hand side. And you can see on the y-axis, we have the number of times we see that fragment size in each of the disease entities. So healthy is in blue, plexiform neurofibroma is in black, MPNST is in red. MPNSTs are towards the top on the left-hand side. So we see shorter fragments more often in MPNST. Plexiform neurofibroma, again, we see that more often than in healthy individuals to be short, but not as much as MPNST. And then healthies, you can see shift as we get in longer fragments, and those become the more predominant fragments in the 150 plus range. And So that is what we used as our in silico size selection in the first papers to say that, okay, fine, let's make that haystack a little bit smaller. I'm trying to find MPNSTs. I'm just going to look between 90 and 150 base pairs. I know that that's mostly where it's going to live based on that work. But this also led us to question whether there's more fine-grained details of these fragmentomic features that we can use to further improve our accuracy distinguishing plexiform from MPNST and potentially apply to atypical neurofibroma to separate those from those two entities. And so our current study, we have patients with plexiform neurofibroma, atypical neurofibromas, and MPNST, as well as healthy volunteers. In each of these patients, we're again collecting blood, we're taking their cell-free DNA, and we're sequencing it. We're taking tumor when available. But here, instead of looking at copy number alterations, we're looking at different characteristics of those fragments. So on the top, in the middle column, you can see I say former end motifs. All I'm talking about there is that each of these fragments has, so DNA is made up of different bases. Those bases, those building blocks, occur in different sequences. And we can look at those sequences at the end of these fragments, and that might be able to separate these entities. That second one, where it says binwise fragmentomics, here in the first paper, again, we're just looking at the average of the cell-free DNA fragment size. But each of those fragments of DNA maps to a specific place in the genome. So if we were to map all of those fragments to the genome, are there certain places on the genome that we see shorter fragments versus longer fragments? And is that different between different disease states? And then the final one is using mathematical modeling, something called non-negative matrix factorization, to try to deconvolute what exactly is in that tube of blood. You might remember I said cell-free DNA it's a mix of normal tissue and then a tumor tissue. So can we tease apart those signatures that are in that tube of blood? And so this first figure is, I agree, not that interpretable, but let me guide you through it. So this is using something called the Delphi method to look at genomic location. So this is the binwise fragmentomics. So on the x-axis, I realized that it did not appear here, but each of those gray blocks is a different chromosome. On the y-axis is the ratio of short, so fragments less than 150 base pairs, to long, greater than 150 base pairs. And what you can see is those top two blocks, plexiform and atypical neurofibroma, aren't flat, but you can see that going across the genome, there's less variation. But if we look at our MPNSTs, pre-treatment and even on treatment, you see a lot more variability. And had the chromosome shown up, you would have seen that it occurs in areas that we'd expect it to, like chromosome 8, which is the really wobbly one right in the middle. And so using this methodology, we're able to come up with models to try to distinguish different disease states. And so here we have a map of AUCs. And so area under the curve, closer to 1 is better. Closer, greater than 7, I'll use as saying it's okay. People might push me on that a little bit. The ones in the middle are blue because they're not that great. But you can see that we do improve over our copy number alterations. So separating plexiform from healthy. So remember, there's not many copy number alterations in plexiforms. We're able to do that with an AUC of 0.87. With copy number alteration, we're at 0.65, not that much better than a coin flip. Our MPNSTs from all other disease states are significantly better. 0.99 is a really good AUC. But you can see that in that middle, we still leave a lot to desire. So yes, we improved a little bit our ability to separate atypicals from plexes. Our AUC is 0.58 from 0.45, but we can do better than that. So the next step was to think about, okay, what causes these different size fragments? And there's a few different answers to that. Some of it is chromatin availability. So as the chromatin unwinds for transcription, areas that are exposed are more likely to be cut shorter. They're more susceptible. But another part of it is that the things that cut those shorters, the nucleases, the scissors, those are different in different tissue types. Not only are they different in different tissue types, they're different between cancer and normal. And so nucleases, those scissors, also have specific sequences, specific base pairs that they prefer. And so on the right, you can see we do an ad hoc analysis of bulk RNA data from MPNSTs and normal tissue, and there is differences in nuclease expression between the two. So if we look at those in motifs, are we able to separate between these disease states? This is a heat map, which doesn't show you much other than to say that on the right, we have all of our different N motifs. You can see that on the left, we're grouped by disease type. And there are some differences between MPNST atypicals and plexiform neurofibroma. And this didn't project at all, but you'll have to trust me or ask me questions if you don't trust me, that what this does is it shows differences that there are certain N motifs, so there's sequences of 4 that occur more commonly in plexiform, in all peripheral nerve sheath tumors than normal tissue. And there's also specific ones that are more common in plexiform, atypical, and MPNST. So taking that all together, you can see that we improve our ability to distinguish between atypical and plexes. So we go up to 0.74 relative to 0.58 on our Delphi method, as well as atypical and healthy, but we see some fall off in our ability to distinguish MPNSTs. So to try to tie all this together, we turn to non-negative matrix factorization. And again, the idea here is that you're basically just saying that there's a bunch of fragments in there. Are there certain signatures? Can we tease that apart into different signatures representing different disease states, normal and tumor? And so in the middle column, you can see an example where we take that one single curve and we separate it into healthy on the right and cancer on the left, and we see two distinct signatures. And importantly, when we look at samples that have higher signature 2, the cancer signature, that correlates with increased tumor fraction. So that seems to suggest that this is in fact coming from cancer, although obviously more validation is required there. In using NMF, you can see that we are able to separate all of our disease states with relatively good AUCs. And so together, we've defined that copy number this is moving on its own. Copy number alterations accurately distinguish malignancy from benign plexiform neurofibroma, that there's distinct profiles in fragmentomics. And I'll skip to this because this is the most important, but we can talk more about that. But we can use bin-wise as well as nucleosome footprinting, as well as in motifs to separate those disease states and potentially putting all of those together in a multimodal model. And then the perhaps most important slide is a plug for NIH's Camp Fantastic. So this is a camp for patients with cancer and cancer survivors. There are also camps for patients, family members of patients. So we've been on a hiatus for the last few years. We're back on this this summer, and I'd encourage any families to participate or providers to refer their patients. So with that, thank you to the large group. This is a collaboration with Angie Herbie and Adil Chanduri at WashU St. Louis. Brigitte and Jack Shern are my mentors at the NCI. And thank you again to the gracious funding from CCF.
Okay, so questions. A couple of questions. I'll start in the back there with Ashley. Oh, wait, sorry.
Hi, thank
Jamie.
you. So I'm curious, I know in the adult solid tumor space, we've seen that certain tumors shed more DNA, and sarcoma is generally one that we think of as being more quiet. So have you had to do something on a technical level to be able to obtain enough DNA to actually do this analysis?
You're exactly right. So in the adult world, so GI cancers shed DNA like crazy.
Right.
Those are beautiful models to use and to validate everything on. But you're right, sarcomas don't tend to shed as much. We do see some differences both in terms of location and our cell-free DNA yield, but a lot of it does come with pre-processing. And so we do some manipulation at the beginning to try to improve our cell-free DNA concentrations, but that is a limiting factor. We don't have a lot of template that we're looking at, and so we try to use only one tube of blood, but sometimes that's difficult.
Okay, super, thanks.
Yeah.
Ashley.
Thank you. I was just wondering, so if this works and you can identify early on that you have this malignant tumor, is there any way that you can identify where that cell-free DNA came from? I know that it's just floating around, but is that do you just then rely on other clinical methods to find the tumor?
So it's a great question. So the short answer is I imagine that at the beginning, if this were to roll into clinic, we would rely on imaging. This would clue you in to say, hey, we can get insurance to pay for a whole body MRI now because our index of suspicion is much higher. With that said, my dream would be that there are different signatures that we can see in here that I didn't really get into, but that we're actively investigating. So the methylation profiles vary by tissue, and you can also see, so from tissue that's near the tumor, you can see inflammation, and you can see those markers there as well. So we can look at methylation either by doing methylation sequencing of the cell-free DNA, which we've done. It's easy, it's expensive though. But another thing that we can do is those in motifs are actually kind of specific to methylation. And there's some modeling that you can do where you can look at places that are methylated or unmethylated. We know are differentially methylated regions from tissue projects. And so we can use that to kind of tease apart the tissue of origin. So Cancer-Seq is probably the best example of this in the adult world, but I think we can do it in pediatrics as well.
Thank you.
On that side? I saw somebody raise their hand over there. No? Okay. Do the microphone up here.
I'm sure you've seen in adults there's some confusion caused by clonal hematopoiesis. And with respect to copy number and the other parameters you're looking at, is this going to be a problem?
So it's complicated. So the short answer is in most children, no. The patients we're looking at aren't necessarily most children, though. So for everybody, clonal hematopoiesis, as your body matures, you are going to just have random mutations that there's debate as to whether they're likely to cause cancer decades later, but for the most part aren't going to cause cancer right then. And this is normal, but it happens to occur in a lot of genes that normally would flag our interest and say, okay, this is really concerning, this is associated with cancer. This happens accumulatively over a lifespan, so we don't see it a lot in pediatrics. Things that increase that, although there's specific chips that are associated with it, which is our acronym, because we have to have acronyms for everything, are receiving chemotherapy, so MPNST survivors who are still at a higher risk of receiving more MPNST. And there's some evidence, and this will hopefully be a future paper, maybe funded by CCF, that in cancer predispositions, there's specifically an increase in chips, and they tend to, again, be in certain genes. So we do have germline for all of these patients, so we take white blood cells at the same time, and so we're able to subtract that out. But yeah, consideration, like a few years ago, I would say no, not for children, but as we sequence deeper and deeper, we see that it is there.
And I thought it would be useful for the group if you explained how you infer copy number.
Yeah, so we can infer copy number. It's complicated. So we use we use something called hidden Markov modeling, but basically the idea is that as we look at the number of reads, so we can see the number of times each base is read, we can look at the rest of the genome and we can normalize to that. And you can see areas that are overrepresented, so there's more reads in these specific regions or fewer reads for deletions, right? And so there's a lot of filtering that you have to do. You can get a lot of false results that way, but that's the basic structure of how we infer copy number alteration. I should also say that there's also exciting nucleosome footprinting, so differences in coverage are not just artifact from your sequencing, it also has to do where those nucleosomes sit, and we know where to look for those nucleosomes, and so we can potentially infer transcription from that as well. This works in mice and patient-derived xenografts, and so we can follow mice with patient tumors longitudinally to see how their inferred transcription changes during treatment. And we know resistance is a big issue. A lot of these patients respond to chemotherapy up front, but then develop resistance very early on.
Another question in the middle.
Hey, Rosa.
Taylor, this was beautiful. I just had a quick question. So given that your DNA amount is lower than usual, do you think that the MPNST has to get to a certain size for your level of detection? And is that an aim that you try to follow using maybe correlative MRI, like volumetrics, to see where your lower limit of detection is?
So you were spot on, Rosa. That's a huge challenge. So by copy number alteration, that's absolutely true. So we struggle with tumors that are less than 5 centimeters. That would be the ideal time to identify it, so that's not perfect, but we're continuing to work on that. The shorter answer is that as we add in these fragmentomic data, there's more name tags that we can look for, and that helps us improve our ability to go down. And we've done some in silico dilutions that we can see that we can go down to a lot lower than we can with just copy number alteration, so we can get down to the sub 1% of tumor fraction, but we need to improve that. Correlates like MRI. We also have another project using proteomics to go along with this as well, but I do think it's going to have to be bolstered by another signal.
And another question.
Hey, Taylor, great talk. Thank you. Of course, I'm going to ask you a question about brain tumors. We're increasingly relying on methylation profiling for final diagnostics in brain tumors. We have many cases now where the methylation profiling gives us the diagnosis where the histology actually was incorrect, right? or the initial diagnosis on histology didn't match up with the methylation profiling. We go with what the methylation profiling tells us. So, we also have a lot of tumors where, we either can't biopsy it because of location or other factors, or the only biopsy we can get is a very tiny needle biopsy, and we often can't get enough material to get adequate genetic profiling. So my question to you is, do you envision that this, like a cell-free DNA technique potentially from CSF, could help either bolster or even in some cases replace or be a secondary measure for us to get the same information? Or do you think that we're not going to be able to get that level of detail?
I'm glad you mentioned by CSF. That's a much easier answer. So by CSF, yes, we can detect it, and there's good studies showing that. We have a study actually with the Pediatric Brain Tumor Consortium looking at some of this as well. From plasma, maybe. It's difficult with these larger genome-wide screens, but when we go really deep, so if we use panels, we can see it. That obviously requires some a priori knowledge. So you have to know what mutations you're looking for, which maybe works in a clinic, but it'd be great to be able to look at least from a research standpoint, more wider nets, and we can't do that very well from plasma for brain tumors, just because a lot of it is filtered out. But more proximal to the tumor, better. We can get it. We can get the cell-free DNA that way.
And a practical question: what's the turnaround time so like you get a sample from a patient? What's the turnaround time to get the analysis?
From me, so there's a lot of people in this room that know that I'm not necessarily the quickest at turning things around. So for me,
this was in a medical laboratory.
In a medical laboratory, so it's easy sequencing. So the cell-free DNA isolation itself takes about 30 minutes. The sequencing, it depends, but especially if you're going relatively shallow, we could turn that around in a few days. With that said, you have to have a lab that has space to get it in a few days. And so really the limitation is both cost and sequencing space. Research, I usually turn these around in about 6 weeks, but
Cool. Thanks.
Great. Well, thank you very much, Taylor. It's really a good talk, and congratulations on all the work. Yeah.